Okay, so welcome to this next video in which we are discussing ELIC and GLIC channels. Okay, so we've now discussed the structure of these cis-loop ligand-gated ion channels found in humans. Let's now discuss the structure of these two new channels found in bacteria, namely ELIC and GLIC. So we'll start with ELIC. Now ELIC is found in a bacterium known as Erwinia chrysanthemi. Erwinia chrysanthemi. Okay, and chrysanthemi is spelt like this. However, this bacterium, Erwinia chrysanthemi, has been renamed recently. Its new name is Dicea dadantii. Okay, Dicea dadantii, I think, anyway, something like that. Erwinia chrysanthemi has been renamed Dicea dadantii, okay? And in this bacterium, it has this ELIC channel, and ELIC stands for Erwinia, for the bacterial name here, Erwinia ligand-gated ion channel. So the LIC is for ligand-gated ion channel. And basically, this thing has a very similar structure to the cis-loop ligand-gated ion channels that we see in humans. Okay, so this means that it's become of huge interest to people studying these cis-loop ligand-gated ion channels. And indeed, we have been able to elucidate much about these cis-loop ligand-gated ion channels by studying ELIC in Erwinia chrysanthemi, or Dicea dadantii, as its new name is. Okay, right. So, let me discuss what the structure of ELIC is then. So, ELIC, it's a pentamer again. So, if we show its structure here, it is a pentamer. So, it has 5 subunits making up the entire ELIC channel. So 1, 2, 3, 4, 5. Okay. So here it is a pentamer, and it's in this bacterial cell membrane. And by the way, Erwinia chrysanthemi is a gram-negative bacillus. So gram-negative bacteria will have 2 cell membranes, an inner cell membrane and an outer cell membrane. So let me show this. So this is a gram-negative bacillus, or a rod. Okay, so it's gram-negative, which means that it has 2 cell membranes, an inner cell membrane and an outer cell membrane, with a very thin cell wall in between them. So let me show this very thin cell wall. I'll show this in red, since it's the cell wall that will be stained in the gram staining process. Okay, so here's the very thin cell wall between the inner and outer membrane. So this is the inner membrane of Erwinia chrysanthemi, and then the membrane outside the cell wall, that's the outer membrane. Okay, so you find these pentameric Erwinia ligand-gated ion channels, ELIC channels, in the inner membrane of Erwinia chrysanthemi. Right, okay, so they are a pentamer, so they're sitting in the cell membrane like so. And if we now take one of these subunits out of the ELIC channel and look at its structure, then it has an almost identical structure to the subunit structure of our cis-loop ligand-gated ion channels. So if this is our phospholipid bilayer here, then what we find is something that looks like this. Okay, so here's the C-terminus, and here's the amino terminus. Now, what do you notice is missing? The cis loop is missing. There is no cis loop on the subunits that make up the Erwinia ligand-gated ion channel, the ELIC channel. Okay, so that's the one difference that they have compared to our cis loop ligand-gated ion channels. That's their big difference. They still have the 4 membrane-spanning domains named M1, M2, M3, and M4. They still have this intracellular loop between the M3 and M4 membrane-spanning alpha helices, known as the M3-M4 loop. And they still form pentamers, just like the cis-loop ligand-gated ion channels in humans. Okay, so what is the ligand which opens the ELIC channel? Well, basically there are two ligands. Now, one is very familiar to us from human biology. One is the molecule GABA, the main inhibitory neurotransmitter within the brain. Now, what does GABA stand for? Well, GABA stands for gamma amino butyric acid. Okay, so let me show you the structure of that because it's very nice and simple. So, butyric acid is the old name for butanoic acid. So you've got a 4-carbon carboxylic acid here. So here's the carboxylic acid group, and then add 3 more carbons to get the number up to 4. Then add hydrogens onto all of these carbons, like so. And then finally, you need an amino group of the gamma carbon. So what is the gamma carbon? Well, the first carbon off the carboxylic acid group is the alpha carbon, and then you keep counting beta, gamma. So this is the gamma carbon here. So the amino group is off this gamma carbon. So this is the structure of gamma amino butyric acid. And gamma amino butyric acid, GABA, is an agonist for these ELIC channels. So it will bind and it will cause the ELIC channel to open. Okay, but ELIC also has another agonist. Okay, so the other agonist is a molecule called cysteamine. Okay, so this is cysteamine. And the proper chemist name for cysteamine is to call it 2-amino-ethane-thiol. So 2-amino-ethane-thiol, and I'll show you exactly why it's called that in a moment. Okay, right. So why is it called cysteamine to start with? Well, basically it's called cysteamine because it's based on the amino acid cysteine. So let's redraw our amino acid cysteine out and then see what modification you have to make to cysteine to make cysteamine. So here's the core amino acid structure, the alpha carbon, the carboxylic and amino acid groups. Okay, so then in cysteine, the R group that you have is a methylene group, and then with a thiol group off it there. Now to convert this into cysteamine, so this is cysteine, to convert it into cysteamine, what you need is, here's the amino group, okay, still here. So actually I'll draw it exactly as it is. So here's the alpha carbon again, and you just take off this carboxylic acid group and replace it with a hydrogen. Okay, so that's how you go from cysteine to cysteamine, just by removing the carboxylic acid group of the amino acid cysteine. Okay, and the reason this is called 2-amino-ethane-thiol should be pretty obvious. Ethane is the 2-carbon carboxylic acid, so just a little bit of revision of organic chemistry here. Okay, so the 2-carbon fully saturated hydrocarbon. Sorry, did I say carboxylic acid? That's terrible. No, the 2-carbon hydrocarbon, which is fully saturated, is called ethane. Okay, then we can go to ethane-thiol, and basically ethane-thiol just means ethane with a thiol group stuck on the end. It's like a alcohol, but instead you have a thiol group rather than the normal alcohol group. So here's the ethane-thiol molecule, and then this carbon will be called the first carbon of the ethane-thiol. This carbon will be called the second carbon, okay? And then you just want to add an amino group onto the second carbon to make 2-amino-ethane-thiol. So that name is perfectly sensible. Right, so cysteamine is also a ligand for the ELIC channel. Okay, right. So now let's turn our attention to the GLIC channel. So GLIC then. GLIC is found in a bacterium known as Glioblastoma violaceus. Okay, so Glioblastoma violaceus. Okay, right. And this is a cyanobacteria, and basically it has this GLIC channel, and GLIC, you might be able to guess now from what ELIC stood for. GLIC stands for Glioblastoma ligand gated ion channel. Okay, so GLIC is the Glioblastobacter ligand-gated ion channel, although we're going to see that it isn't actually a ligand-gated ion channel. It is in fact gated by pH rather than by a ligand as such. Okay, right, well I suppose you could call it a ligand because of course pH is proton concentration really, so it's gated by protons. Okay, but it's not a conventional ligand. Right, so let's look at the structure of the GLIC channel then. Right, so again the GLIC channel is a pentamer. So it will sit in the membrane of the Glioblastobacter violaceus. So here it is. Here's the pore down the middle. And then it is divided up into these 5 separate subunits here. Okay. And now if we take one of these subunits out of the pentamer and look at its membrane-spanning topology, then we're going to see 2 changes from the cis-loop ligand-gated ion channels. So the first is that you have no cis-loop in the GLIC channel, so no cis-loop again, and you also have no large intracellular loop, basically. You have no large M3, M4 loop anymore. So the important alterations are no cis-loop in the extracellular domain, okay, and also no intracellular loop, no M3, M4 loop. Okay, so you take 5 of these GLIC subunits, pentamerize them together to make a GLIC channel, and that's what happens in the Glioblastobacter violaceus. Right, okay, so this channel is gated by pH. So here's the channel sitting in the phospholipid bilayer. And basically, if the pH intracellularly goes down, okay, so if pH intracellularly goes down, now remember what pH means. It is negative log to base 10, okay, of the proton concentration. Right, so if we plot this as a graph, okay, so let me show you this. So if you have the x-axis here and the y-axis here, and you plot y is equal to log to base 10, firstly, of x. So you're taking every number along the real axis and you're going to plug it into log to base 10. You're going to ask what number would you have to raise 10 to get x. Basically, y is going to be the answer to this question. 10 to the power of what is equal to x? That's what this function is going to spit out for you. So you can't do it if x is a negative number. It just doesn't work. Not in real analysis anyway. In the complex numbers it works. You can define a complex value for the logarithm of a negative number, but in real analysis we're working in the real numbers. We're not working in the complex numbers. Okay, so so we can only define this for the positive real numbers, but basically if you do it, if you put in a number like 0.01, so for instance, like, let's do a few examples so that we can get the gist of why this works. So if you put in it, for example, log to base 10 of a tenth, okay? So what number would you have to raise 10 to get 1/10? Well, that's negative 1. So 1/10 here is going to be mapped onto negative 1 down there. Okay, then let's put in the number, 1. What about log to base 10 of 1? So if we put in 1, what are we going to get? So what do you have to raise 10 to get 1? Well, anything to the power of 0 is 1. So the value that 1 should be mapped onto is 0. And then let's put in something like 100. If we put in log to base 10 of 100, then we're asking what power should we have to raise 10 to get 100? Well, that's 2. You squared 10 to get 2. So if we put in 100, you're going to go to 2. So over here where 100 is, you'll have 2. So basically this graph is going to look something like that. Okay, so this is the graph y is equal to log to base 10 of x. Now, basically if we take y is equal to negative log to base 10 of x, what instead you'll get is something that looks like this, the whole thing flipped in the y-axis, because every number that you were mapping x onto, so every x value in the positive real line was being mapped onto a y value, but now it's being mapped onto the negative of that, so it's just going to flip every value that you were mapping each x value onto. It's going to flip that in the x-axis, basically. So the graph you're going to end up with is going to be this turquoise graph now, here. So, now just imagine that x is proton concentration. So on the x-axis we're plotting proton concentration, and on the y-axis we're now plotting pH. Okay? Now, it doesn't matter to us that the graph isn't defined for negative concentrations of protons, because there is no such thing as negative concentrations of protons. So maths isn't limiting us there. Okay, and you'll see that as the proton concentration goes up and up and up, the pH gets smaller and smaller, and as the proton concentration gets smaller and smaller, pH goes up. So basically, as proton concentration gets bigger, pH goes down. So, at lower pHs, you'll have a higher proton concentration. Okay, so when pH is low on the intracellular aspect of this receptor, that means that proton concentration is high. That's what causes the GLIC channel to open. Okay, so that's a basic introduction of ELIC and GLIC.